Fusion, the international science radio show.
My name is Dipon Sarkar, and I'm a PhD student at the Tasmanian Institute of Agriculture here in the University of Tasmania. I'm studying food microbiology, and I'm also an avid enthusiast of science communication.
Were you interested previously in food first or microbiology first?
Food first, for sure. The interest in food began when I couldn't even read or write, I think. And microbiology later. I was always interested in science and life sciences in particular. But then in uni, I got interested in microbiology, and then I was given the option of studying the microbiology of the environment, the microbiology of the human system, or the microbiology of food. And I chose food because I thought that this would let me eat a lot.
And did it?
Not really. Because the stuff I study in microbiology makes the food uneatable. So,
so you find out what is safe to eat.
Yes. So the specific field that I'm researching on is called food safety, where I study how pathogens grow in cheese, how fast they grow or how slow they grow. So those cheese that I study in the lab are a big no-no for eating.
And you're particularly looking at paneer.
Yeah. So my research matrix, or the food of interest that I'm studying for my thesis, is paneer. It's a fresh, soft cheese that originates in India. It originated there, but now it's produced and consumed everywhere in the world. Here in Australia, we have at least 5 brands that's making paneer, and a lot of people making it at home. And I'm studying how pathogens would grow in paneer if it's contaminated, because although it's a very old product, not a lot is known about how pathogens behave in the product.
And my understanding is that unlike some of the cheeses that are produced with microorganisms, paneer isn't.
No, so paneer is a fresh cheese. So fresh cheese means that it does not have any other microorganisms in it. So we don't intentionally put a fermenting or a starter culture in it. One of the inherent advantages of having a mature cheese is that there's already microorganism present there that you have intentionally put. And these microorganisms often have a protective effect, and they don't let bad pathogens grow on it, or they protect it a bit. So these fresh cheeses like feta, paneer, queso fresco, these don't have this protective effect from starter cultures. So they are sometimes more prone to pathogenic microorganisms or spoilage microorganisms.
And how do you study this?
So to study it, I basically mimic a contamination incident. So I intentionally contaminate my samples that I make in the lab, and then I study how they grow over time. And this pattern is reproducible. So given that the conditions remain the same, this pattern of growth is very similar. And if you study it enough, you can predict it. So the observations help me make predictive models that are then hopefully would be used by manufacturers and food safety regulators to make decisions.
And you've been involved with the Young Tassie Scientists and Science Week. What have you been up to?
Young Tassie Scientist and Science Week is now the highlight of my year, to be honest. August is such a busy time and such an exciting time because of these two things. So Young Tassie Scientist is, if your viewers are not aware of it, it's a science outreach program that is run through National Science Week and University of Tasmania, where there's a group of scientists that forms a cohort, and we go into schools. Normally, when in the olden days, we used to physically go to a school and talk to students about our research or about science in general. But now like everyone else, we also shifted to a Zoom platform. So this year we we have made videos and we circulated to students, and they saw it and they had a bunch of questions ready for us when we entered their classroom. They were talking about our research, our science, what got us interested. And it's very, why I got involved in it is it's very enriching, because not only are they learning, but I think I learn a lot by talking to kids, because they have such an innocent, like the innocence in the questions really makes you think, like, why am I doing what am I doing? What is the whole point of it? So I find it very rewarding to talk to kids about science and even influence them in their career decisions, maybe. And this National Science Week, I was involved in an independent event too, which was called Ferment Home Station. So we applied for a grant and we got one. And the whole event was around talking to people about the science of fermentation. Everyone has been fermenting during this pandemic, like sourdough, kimchi, sauerkraut, so many things. Especially my housemate, he's been fermenting everything. So this idea came in a conversation with him, and we were sitting on the dinner table, and I was like, do you know the science behind fermentation? And we ended up actually applying for this grant together and running this event where we promoted the science behind fermentation.
And so how have you been doing this? Do you have videos?
We have invited experts from around the world. There's been the only sourdough librarian of the world joined us and talked to us about his sourdough initiative in Belgium. Morgan Clemenston is an expert baker based in Sydney. She came and talked about her sourdough journey. Erin McKinney is a fermentation scientist in the University of North Carolina, USA, and she ran a fermentation workshop. And all these events have been recorded, and it's available on the YouTube channel. And they were all live events, so people joined us live. And we also ran a public experiment where we had 50 kids with instructions that would help people make a sourdough starter from scratch, so a wild sourdough. And while making the sourdough, they could record some data and contribute it to a international public science experiment that's already going on. So we sold all the 50 kits, and we are getting some data in. So hopefully soon we'll have a sneak peek into how Tasmanians make sourdough starters, and talk a bit more about it then.
So if people are making sourdough starter at home without getting a starter from someone else, how does that work?
So essentially, the flour that you use for your starter already has most of the microorganisms that your starter will eventually have. So you give water, and you put the flour in the jar, and you give water, and you put it in a nice warm environment, and you basically tell the microorganisms to grow. And they have this nice warm home, they have food, so they start growing, and you have to feed your starter regularly, so you give them food. Hence you promote the growth of the microorganisms, and eventually after 7 days, you'll have a mature starter. So research has shown, there is a lot of talk that the air contributes the microorganism, the water contributes the microorganism. The debate's still out. I am not an expert to comment on that, but this much is known that flour is the major contributor to your sourdough microbiota. So you're basically using what's there already in the flour.
So if you
and sometimes your hands.
So if you provide the flour and the water and a good environment, the yeast will come from somewhere in your house.
The yeast is already there in the flour. Those yeast will start growing, and the lactic acid bacteria will start growing. and then they together make this yummy, very tarty environment where other microorganisms will not be able to grow. So they make this, they they have this source of food, and they make this environment so that they can protect their own source, and so other microorganisms won't come and grow on it. That's why it's very hard for your sourdough starter to go like spoil and mold to grow on it. Although you can, like, believe me, you can very easily just leave it there for a month and see what grows. But if you maintain it, it's a self-sustaining environment of where yeast and lactic acid bacteria protect themselves and give you this yummy food.
Amazing. Are there any tricks to making your bread become fluffier with the right things to feed the yeast, or the right way to rise the bread?
I think fluffiness, as far as my limited knowledge goes, depends a lot on the gluten content of the flour. So high gluten breads are easy, fluffs up easier. That's why white flour bread is so fluffy. And like ancient flours like rye and spelt don't rise that much generally. But there's all, like, bakers are amazing at their job, and they've figured out how to make the bread physically, how to knead it or how to proof it so that they can get the maximum rise out of it.
And with food spoilage, are there general rules that we should know about how to look after our food so that it stays edible as long as possible, or so we don't hurt ourselves?
Put it in the fridge.
How long?
So temperature is known to be one of the most essential controls of food. That's why essentially with the discovery of refrigeration, our food habits changed. So before refrigeration, it was something else, and fermentation was a big thing then, because fermentation is a natural way of preserving food. So we have preservatives, both chemical and natural, that helps the food last longer. Generally, less exposure to air and heat helps food stay non-spoiled for a longer time. And if there's special considerations for your food, for example, if you're fermenting, you should ensure that, like for your sauerkraut, all your cabbage should be under the liquid. So under the liquid is an anaerobic environment where they're not getting oxygen. Whereas if they go above the liquid and if they have access to air, they will spoil for sure. No matter how much you're fermenting down on the liquid side, mold will be growing on the exposed part. So if there's special consideration for your food, follow that. Otherwise, fridge and freezer.
But are there's definitely limits on how long you can keep things in the fridge or the freezer? For
sure. Like, forget your pasta in the fridge for a month and see what grows on it. So that is what, because when we heat, we kill things, but then we leave it open to cool, or we are eating it. So we are exposing it to a lot of microorganisms, and a lot of it are spoilage microorganisms, molds, fungi, and then we wrap up our leftovers or put it in a Tupperware and put it in the fridge. So the microorganisms and the molds are already there. The fridge really slows them down. Like, it's like what happens to people in winter. They get like all shrivelly and slow. So that's what happens to microorganisms. They become slow essentially. But slow doesn't mean death. So they are growing very slowly, and eventually, if you give enough time, they will outgrow it, and you can see more. That's why they spoil even in the fridge.
And I guess even in the freezer.
Even in the freezer, yeah, sometimes. Another reason why we did this ferment home station event now is not only are people fermenting a lot, but also at a time when a microorganism is basically changing our world. There's a lot of focus on hand washing and personal hygiene to kill microorganisms. But I think the narrative should be a bit more equitable, in the sense that there's also wonderful microbes out there giving us food, antibiotics, helping us give our coffee, our jeans that we wear. So something that young Tassie scientist also has like put to me is that kids especially are aware of microbes as germs, and you and yak are the things that associated with microorganisms. But what about this amazing world that they give us too? Antibiotics. We won't have antibiotics unless we had microorganisms. So, um, so this event was focused on talking about the good microbes for a change, because a bad microbe is really changing our world.
Absolutely. And chocolate is fermented before it's roasted.
Yeah. Chocolate, coffee, genes, antibiotics, so many things. Yeast is like bread, beer, wine, sake, all the things that we like to enjoy in our evenings. So
Absolutely. And have you been involved in other science communication?
Not really. Young Tassie Scientist is an annual program that I'm involved with. So it happens every year. I've done it 2 years now. And also as a part of Young Tassie Scientist, we do other events, like we go to schools otherwise, and we do events like Tastrofest and AgFest in Tasmania. So those are the communication programs I'm involved with. But I'd definitely like to be involved more. And yeah.
And so you've got a story about yeast?
Yeah. This story about yeast is basically the history of baking. And I heard this on a podcast. And it was like a mind-blowing moment for me, because even though it's all around us, I don't think a lot of us would be aware of it. I certainly wasn't. Is this paketed yeast, the instant yeast that was out of shelves during the pandemic, that made people make sourdough? This yeast has not been around for a long time. Like Louis Pasteur discovered it, I think, in the 1860s. And it wasn't industrially produced and packaged until like the the early 1900s. But we've been baking for thousands of years. Like Egyptians were baking, so they didn't have this packaged yeast. How did they bake? Because they had figured out sourdough many years ago, and if not sourdough, some other way of harnessing natural yeast. And in the 1800s, like the bakers were called that they were slaves to their starters, because they had to feed it every hour. They couldn't sleep, like, because they had to wake up like every 4 hours to feed it. And when instant yeast was discovered, that changed like baking. That was a major disruption in the baking industry, because people realized that we don't have to do all these things. This packet can just cause wonders. So a lot of countries actually lost the traditional history of making bread, like completely lost it. So sourdoughs were not there. Only the countries that produced like ancient grains like rye kept then ancient knowledge, because they were seeing that yeast didn't affect rye that well. So that is because there's a compound called phytic acid in the grains, and rye has a lot of phytic acid. And yeast cannot break down phytic acid, and neither can our human body. So we eat it without digesting it. And phytic acid is not only is it undigestible, it also holds on to, it's a something called a chelator. So it holds on to other things, other micronutrients, and doesn't let our body absorb that too. So the yeast bread that we sometimes eat, we're not digesting a lot of the nutrients from it. The wonderful thing about sourdough is not only is there yeast, there's also lactic acid bacteria. And lactic acid bacteria can actually break down this phytic acid for us. So even before we eat it, this acid is broken down and these micronutrients are now available. And that's when we eat it, we really digest all of it. So the amount of nutrition that we're getting from like sourdough bread is much more than yeast bread.
Oh, and that uh, sourdough starter library in Belgium, can people make
Yeah.
withdrawals?
Sorry.
Can people make withdrawals?
It's not a library where you can rent the books there. It's somewhere where you submit your starter if you have a very unique starter, so that it doesn't get lost with time. And basically, if I had a unique starter and I don't have family or friends who wants to take care of it, uh, there's people out there who really want your starter and would like to preserve it. So there's amazing stories. Check out their website, and they've got a Quest for Sourdough website where there's some 200-year-old starters there. So
Do you remember what it's called?
Quest for Sourdough.
Quest for Sourdough. All right, I'll put a link up on the show notes for this episode.
Yeah.
Terrific. Well, that's really interesting. And just thinking of the bakers getting up every hour to feed their starter.
Honestly speaking, that I didn't know that, and I was listening to a podcast, and I was like, I've never thought of that.
And no fridges.
Yeah, no fridges, no instant yeast. How did they bake? Yeah.
Amazing. Do you have advice for students who are thinking about studying microbiology?
Um, don't be limited by what you see. There's a huge world under the microscope, and they do amazing things and also horrible things. So you can study either, and either way you're going to be helping human society anyways. So um, so dive in, and like there's a wonderful world there that we aren't always aware of because we can't see it.
Well, Dipon, thank you very much.
Thank you.